Okay, today we're gonna talk about ChIP-seq. So let's first do a recap. We still have a little unfinished business from the last lecture. We know that genes are regulated because transcription factors recognize specific sequence patterns to bind to the DNA, then it can regulate nearby gene expression. And in order to find those transcription factors and where they bind and figure out, what's the effect they have on the nearby gene, we, in the last lecture, talk about the different computational approaches for motif finding. These methods are okay when you look at the yeast or bacteria, and maybe even C. elegans. But by the time you look at human mouse, things are usually quite complicated. We mentioned that regulatory sequence that are bound by transcription factors, it can be quite far away from the target gene, and the sequence can be short or long. The motif can be short or long, and you can already start to see them appear in combinations with other transcription factors or clusters of the same transcription factor repeating itself, just so that the transcription factor will sure to bind that location and regulate the nearby genes more tightly. In most of the case, we don't have enough of these gene expression perturbations to give you a very good set of genes that are co-expressed. By the way, the genes that are co-expressed in a cluster, we hypothesize that they may be co-regulated, but they are not always the case. Sometimes when the signal is here, this transcription factor regulates something, but one of its child also is a transcription factor and they regulate other things. So you would see the uncles and together with the children together, but you hypothesize that they share some similar signals. And so in order to do motif finding in higher eukaryotes, people look at, first of all, over the last 20 years, there are more and more known transcription factor motifs that are experimentally derived. People start to look at comparative genomics to look at sequences that are conserved. They start to look at the motif modules for motifs that are clustered within a short range of each other. And then they do ChIP-ChIP or ChIP-seq. So let's go over each of them. PBM, we went through last round, so there are experimental approaches to get the motif information for some transcription factors. And one thing that people notice is that usually the same family of transcription factor, say GATA1, GATA2, GATA3, GATA4, GATA5, they very often recognize quite similar transcription factor motifs. They might be expressed in very different cells. They might also interact with different partners and regulate different target genes, but the specific sequence pattern they themselves recognize can be identical or quite similar. With comparative genomics, this is something that people do. So UC Santa Cruz is the genome resource. When the first human genome was sequenced, there needs to be a genome browser for people to download the different gene annotations, to visualize the sequence, to look at how genes reside in the genome. And so that's resided in UC Santa Cruz. And they have developed this tool called phase conservation score. Basically, they look at whether the sequences are under strong evolutionary constraints. Initially, there was like human and mouse, and they're like, now you can see here, this is different vertebrates, mouse, rat, rabbit, dogs, armadillo. In fact, because genome sequencing now is so inexpensive, people are sequencing hundreds or thousands of different species, and you can align the sequences together. Especially, usually you align them by the genes, because genes are still much more similar between the species. And once you can align the genes, you can look at the sequence near in front of the gene, in the introns of those genes, and also after the end of this gene. And you can do this type of multiple sequence alignment. And this is an example of some alignment. You can see these sequences, they appear. So by the way, we're looking at the main track is human, and then you are comparing with mouse, rat, rabbit, and dog, and so on. So if there is a black mark here, it indicates that sequence at least is also present in mouse, and also how similar that is to the human sequence. Whereas in chickens and tetradon, those sequences don't really appear. And so you can see there is a conservation score in here. If it's very high, it means you can see here, this is very deeply conserved, which means this sequence is present and also quite similar in all the other species. Whereas you can see this region is only present in human but doesn't appear in the other species. So it's a not very conserved sequence. So phase conservation score is between 0 and 1. 0 means this is not conserved at all. 1 means it is very, very conserved. But there are different tracks of this phase conservation score because people sometimes are interested in only conservation within all the primate species, orangutan, gorilla, you know, those. And then there are people who are interested in all the conservation within, say, mammals, right? And then you can go all the way to fish, right? So you can download different tracks. They tell you how many species are used or which are the species used to generate the alignment and calculate that conservation. And so this information can be very useful. You can imagine that if we cut off at say 50% conservation, so this, remember, is a score between 0 and 1. If we cut it by 50%, 0.5 on the conservation score, you are not going to eliminate just 50% of the sequence. You can see if you cut off at this point, you are going to eliminate probably 90% of the sequences that are not conserved. That is not to say that the things which are not conserved are completely useless. For example, if you want to look at some genes related to ovarian cancer, and you want this thing to be completely conserved in fish, it's going to be harder because the fish does not have an ovary. Well, no, actually they do have egg, but it's, or breast, or, you know, like, you know, depending on the level of regulation, you may or may not need the conservation to be that deep. Sorry, fish does have ovary. They don't have a mammary gland, right? So that's the difference. And so you can use conservation to eliminate sequence so that if you have to look far away from the transcription start site, you don't need to look at all the sequences. You can narrow down your search space. So there are some very interesting studies generated on this. For example, this is a region in the genome that's a gene desert, which means the nearby genes are really, really far away. And suddenly there's a region, there are actually islands of these little regions that are a few hundred base pair long. And if you look at their conservation, from mouse or so from human to mouse to chicken, frog, fish, it's really ultra conserved in all of the vertebrate species. So I want to ask you some question. Is conserved sequence functional? If you have something like this that's really conserved, if it is this conserved, it's got to be functional. Just because we don't know what they do doesn't mean that they are not functional. Yeah, so there's a reason for this thing to be ultra conserved, right? And so yeah, so some of them of these conserved regions are in the exons enriched in RNA sequence processing. Those are in the coding regions of the RNA. And sometimes it's important for splicing, but the really interesting thing are sometimes even in gene desert, there's in the middle of nowhere, right? That suddenly you see this region that's really conserved in the species. And then there are some experimental work that are conducted to test this. So we mentioned that conservation does not mean function. If something is not conserved, it can still have function. But if something is super conserved, it's got to have function. And this is some example that people have done. Interestingly, the sequences that are super conserved throughout the species, they are usually related to development, because, you know, when we are embryos, whether it's a pig or a rat or a human, they all kind of look like little fish, right? So those early development genes regulation has to be tightly controlled, and those are very, very conserved, and you do see that happen. And so, for example, in order to see what this region does, you know, you could insert this into or label the genes near this region and see. So you put a gene, like a reporter gene, that give out a blue marker, and you ask when is that gene getting activated and potentially regulated by the sequence. So you can put a blue marker gene near that region and see whether you have this sequence or you delete this 200 base pair sequence, that marker gene will be different. And so in the embryos, you might be able to see, oh, this appears in the brain or in some heart of the early embryos. That will give you some idea. Roughly what tissue is this region important, and you know what, and to really show that it has a functional role. So you can mutate this in different ways and see whether that changes the phenotype. Okay. And another interesting study that people have looked at is human accelerated region. So there are regions in the genome, for example, in this example, this is the human sequence. And if you look at the chimpanzee, rhesus monkey, mouse, rat, dog, chicken, they are quite conserved. And suddenly in human, all these bases are changed. There seems to be a positive selection, meaning that this particular nucleotide, once it changes, somehow giving human a much better evolutionary advantage. And therefore they are being fixed into the genome. And you wonder what they are. And so scientists have also done this type of research. And for example, they found that this particular sequence is actually very important for neurodevelopment because of these basis changes, humans are much, much smaller. In the same study, they have also shown that the accelerated regions, the sum sequence are very different in human, because I guess human use the sum much more differently and much more flexibly, rather than just climbing trees we type. So these are the interesting things. So I would say not conserved doesn't mean not functional, but conserved are definitely functional. And you can use conservation to eliminate some of the random garbage, and so you can enrich for your signals to look for motifs. And then we mentioned, we already see this in yeast. Very often motifs are clustered in a region. Usually the cluster is 150 to 200 base pair long. And suddenly within, you can see what in this particular study, they were looking at a 100 to 2000 base pair long region. But very often those modules, we call them a transcription factor module, is usually one or multiple transcription factors bind in a concentrated way within a couple hundred base pair sequence. And that module is very important. You can use sequence conservation first to eliminate a lot of sequences first, and then within each of those fairly, a little bit more conserved sequence, you can ask, do you see the same motif appearing over and over again? Do you see a combination of different motifs and things like this? And so there is a study generated on this, and they can eliminate a lot of the irrelevant sequence in the genome. And interesting, a lot of the regions they identified later was proven to have very important functional roles. And so this kind of are some landmarks. Suddenly you see a lot of things happening there. That's an important regulation region. And if you do that for motif finding, to look for motif clusters, you are more likely to get some interesting result. But despite all of this, I would say transcription factor motif finding is just not very easy. In fact, when I came here in 2002, as I mentioned, our collaborators just have some mouse experiment when they treat with the mouse with 2 drugs, and then they see 2,000 genes upregulated. And to do motif finding with that, because at that time, the upregulated genes could be many different situations. We don't have a good cluster of genes. It's very, very hard. The savior of my career is this technology, which is called ChIP. Now it's called ChIP-seq. The first ChIP stands for chromatin immunoprecipitation. The second part is now high throughput sequencing. So basically, it's a way for scientists to very quickly look at how a particular transcription factor binds into the genome. At the genome-wide, all the locations you can investigate, and you want to ask, where does this one transcription factor binds throughout the genome? The experiment worked like this. So each of the rectangle here is a sequence on the DNA. Usually we call those cis element. It means the DNA on the genome which is being recognized by the factor. And each of the other shape is a different transcription factor. The red star is one transcription factor. The green circle is a different transcription factor. You can see they are called the trans factor or transcription factors, right? Trans factor just means usually these are proteins and they bind to the cis element in a sequence specific way. And even in naturally occurring, you just grow a cell in a petri dish, or you take cell from some individual tissues, there will be naturally occurring transcription factors that bind to the cis element. And so in the chromatin immunoprecipitation, the very first step you do is to do cross-linking. We use formaldehyde to cross-link the protein-DNA interaction. There's going to be a bond formed between the transcription factor and the DNA covalent bond. So it's very stable. They are now stuck together. And then there are machines that can sonicate, use sonic wave to break the DNA into shorter pieces. Because we have done the cross-linking, the transcription factors are still attached to the DNA that they bind to. And then if we are interested in a particular transcription factor, and if that transcription factor, you have an antibody. In this case, we are interested in this red star protein, and there is already an antibody that has been raised which uniquely recognize the red star protein. You can add the antibody to this solution. You basically, you break up the cell, the DNA, the chromatin is already sheared into small pieces, and you use the antibody to interact with all the red star protein. And this antibody can have a little label at the end that you can spin down with either magnetic bead or something, right? So anything that does not interact with antibody can be washed out. Anything that is, say, the antibody is attached to a bead, then you can spin down the bead and they enrich for all the red star protein, as well as the DNA that's enriched, that's bound by it. You do notice that sometimes, so antibody recognition is not 100%. It's probably, well, you can't think it is 100%. It means that it will probably still make a mistake 1 in 1,000. That's already a very, very specific antibody, but because in the genome, in the cell, there are so many other proteins around, occasionally it will recognize some wrong guys in here. And also, sometimes there are some random piece of DNA that just stuck to the bead or stuck to the tube of your experiment. They also get pulled down. Hopefully those sequences are not consistent. By the way, we do this experiment in usually hundreds of thousands of cells. In the early days, we have to do this experiment with usually a million cells. So it does require quite a bit of starting material. And so hopefully those non-specific piece of DNA and also those non-specific binding will not be repeated in the different cells, you know, whereas this antibody binding to the red star protein and binding to this piece of DNA is consistent in most of the cells. We are hoping that these random noise will, you know, won't be consistent. And after we have enriched for this, there's a reverse cross-linking. You usually just heat up you boil this solution, and then the protein will break off from the DNA. And then you isolate the DNA, and at this point, you can amplify it a little bit, then you can do high throughput sequencing. Okay, so with RNA-seq, you can do RNA-seq with very few cells because each piece of DNA is able to produce a lot of different copies of the RNA. Whereas for ChIP-seq, because you are using a protein to bind to DNA and every cell just only have 2 chromosomes. And so you do need a significant amount of starting material to do this experiment. And so at this point, there would be PCR amplification and then sequencing adapter, then you put it in high throughput sequencing. This is coming from a different review paper. They also show, you know, proteins binding to the DNA, you sonicate into small pieces, and you use the antibody to recognize the protein of interest. You enrich for those DNA bound to that protein of interest, and then you try to do high throughput sequencing. So this is what the signal looks like. Because in different cells, as you use the sonic wave to break up the DNA, supposedly this is one region where there is a transcription factor motif and the transcription factor is binding to this location. In many different cells, the transcription factors are binding to the same, say, chromosome 1 location 10,000. But then when you try to sonicate the DNA into small pieces, the breakage location is kind of random. So sometimes it breaks like here, another time it breaks like that, another time you break that like this. And at the end, when you sequence, by the way, early day when people do ChIP-seq, sequencing is quite expensive at that time. By the way, high throughput sequencing for Illumina, the very first paper published was not on RNA-seq, it was on ChIP-seq, because it was just the count that's important. And at that time, it was, I think, only sequence of 30 or maybe even 25 nucleotide for each fragment. And they can only sequence one end because sequence the other end is twice as expensive. And they just, okay, randomly pick one end to sequence. And the one thing they notice is that you probably see here, if you sequence from this end, remember DNA is double-stranded. If you sequence from this side of the binding side, then it will map to the Watson strand versus the Crick strand, right? So if you sequence from this side of the binding, the sequence will come from the Watson strand. Whereas if you sequence from the right side of the transcription factor binding, you will sequence from the Crick side, right? So you will see basically when you look at many different cells, we're sequencing all of the cells together, right? What you will see is that in some regions, suddenly you will have kind of like a peak or even two peaks. Half of those are coming from the left and they all map to the plus strand on the DNA. Whereas the right side, it's all mapped to the minus strand on the DNA. And very often that means the transcription factor is binding right in the middle, okay? By the way, there is also a control experiment in ChIP, and it's usually using input control. You still do the cross-linking, you still share them into small pieces, but then there is no antibody enrichment. You just directly decross-link and get this whole short fragment, sequence them out into the genome. And you will get an idea if there are some biases, or some regions are easier to sequence than others, you can see that directly. And so that's a control experiment, okay? And so very often, ChIP-seq is done with 1 to 2 chip and 1 to 2 input controls. You can publish even a single ChIP-seq without control if the signal is pretty good. It's not, I would say probably half of the published ChIP-seq data don't even have replicates, which is still okay. All right. Yes, question.
So how do you decide if it's does the transcription bind, how much bases the transcription factor usually bind to? Does it dictate how long?
I see. So the question is, how long are these transcription factors bind? And does that indicate how broad this thing is, right? Different transcription factors bind differently. In mammalian genomes, we usually see transcription factor binding from even 5 to 6 nucleotides to as long as 16, 18 base pair long. Most of them are probably 15, 16 is kind of the rare, is not the norm. The norm is, yeah, I would say 6 to 10. So actually for motif finding, very often we try something like you try a 6, you try a 10, you try a 14, and you just see whether it gives you something. Okay, and so the peak, what kind of shape does it have? It depends a little bit on the transcription factor binding width, but also depends on how harshly you sonicate. If you sonicate for longer time, the fragment will be shorter. But if you don't sonicate as much, the fragments will be longer, then this peak will be a bit broader, right? So in the ChIP days, people don't sonicate as much. They still have fragments that are over 500 base pair because they need many probes to cover that region in order to call it. But nowadays with ChIP-seq, people usually sequence down to about 200 base pair long. When it's too long, when it's too short, sometimes you can't sonicate anymore. Usually, I think even if you were to sonicate like crazy, you probably still see a band around 100. Okay. So that's the ChIP-seq experiment. So now we are going to go through sequence analysis of, sequence analysis of ChIP-seq. You have been in this course for a first module now. If thinking back of what you have to do with RNA-seq, supposedly you have the first ChIP-seq data. What would you do first? Yes, FastQC, right? If the FastQC data is very bad, maybe they should resequence your sample for free. What other things do you need to do? Align the reads, what do you use? Splice aware or non-splice aware? There's no splicing. Yeah, it's directly on the DNA, so you don't use star-seq. You don't use star, you use BWA. Remember first homework. This just is mapping the reads to the genome DNA. And after the mapping, you generate a BAM file. And at that time, there are some, you want to actually look at where this factor binds to. This algorithm was developed in my group back in 2008. And the idea we have was, we realized that because you see there's a transcription factor binding here, and when you break, you usually break to the left and to the right of this binding. You can imagine if you have chopsticks and you put your hands there as a transcription factor binding and you try to chop it, usually it doesn't chop here, right? It can chop nearby regions. And then there's a fragment around here and you're either sequencing from the left side or from the right side. But if you're sequencing from the right side, you are reading from the Watson's, the Crick strand. And so you will see a lot of reads coming from this and a lot of reads on the minor strand like this. And we try to build this double peak. And so at that point, actually, we try to see what is the top, the mode of the blue reads and the mode of the red reads. And usually those distance is roughly the fragment size that you sonicate, or at least the average fragment size that you sonicate. Okay, so that's related to Patrick's question, you know, how broad is this peak? If you don't sonicate enough, those blue peak and red peak will be a little further away, but if you sonicate a lot, they will be close to each other. And because we know the actual binding site is usually right in the middle, and so for each of these original reads, we move them to half of the distance. Because remember, the fragment is this long. We know they're going this way, but if each of the read, we just move it down half of this distance, we're hoping that we're closer to the correct binding site. Okay, and so after we move, you have this, you know, then you can start to look for peaks. And for peak calling, but by the way, MAX was developed before DESeq. And so at that time, we were just thinking, you know, where does the read land on the genome? With like, oh, maybe it's a Poisson distribution. This is before we knew it's negative binomial distribution. We just did Poisson. And remember, Poisson distribution has only one parameter, which is what is your expected read that lands in a, say, 300 base pair region, right? So if you know your total genome size is this and your total read is this, you can ask yourself within a 300 base pair genome, what do you expect? It's a pretty small number, very small number of reads you expect to see in a 300 base pair genome. So one thing we noticed that's kind of interesting is that when you sequence the ChIP-seq sample, also if you sequence the input control, you will notice that there is a very big local bias in the genome. Even people in the early days when they are sequencing the human genome, that's a naked DNA. It's just DNA without any chromatin structure. They already notice that certain regions are easier to sequence than others, for example, GC content. But once you do ChIP-seq, remember we first cross-link, then we sonicate. We start to notice that in the genome, there are some regions that are more exposed. They're kind of, you can imagine a human genome is like a twisted up rubber band. And some regions are more open, and those regions you can sequence easily. And whether you look at the input or the chip, within a few KB region, you see a lot more reads. So we're checking within 10 KB. So we divide up the whole genome, look at a read in 10 KB. You notice some regions, a lot of reads will land there. Some other locations, very few reads will land there. So imagine if the genome is twisted up as a rubber band, you cross-link and you try to kind of sonicate. That little rubber band could be a little ball inside that it's hard to chop up. Right, so those regions are harder to sequence actually. And so understanding this type of bias is very important. Just because in a region you see 5 reads or 20 reads, is this really a peak? You have to look at the control sample to give you an idea whether that region is an enrichment. So supposedly in the chip sample, we see 30 reads land in this 300 base pair region, and in the control, you see 5. Is that a good enough enrichment? 30 to 5 sounds like a pretty good 6-fold enrichment, but what about 6 to 1? Right? That's also a 6-fold enrichment. Do you believe this as a real enrichment or not? Because if you try to estimate this type of local biases with only 300 base pair, you can imagine in the early days when you only sequence the genome for say 20 million reads, within a 300 base pair on average, you only see 1 to 2 reads. So the parameter estimation on the background, on the input control is very, very difficult. And so what we do is we estimate the percent parameter right underneath this chip region that seem to have some reads that are potentially interesting. So this, just say if it's 300 base pair, we also look at 1 KB, we also look at 5 KB, and we look at the 10 KB. In case the estimation on the short region is not robust, we do these 4 different estimations, and also we do whole genome, the whole genome read count. So this is the whole genome background. This is the global background from 10 KB, 5 KB, 1 KB, and 3 KB. And each of them help us estimate a lambda parameter. And at the end, we take the maximum. We use the most conservative approach to estimate the background. And then we ask if using that maximum lambda as the background, are the number of reads we identified from this region significantly enriched compared to the expected background? And so you can imagine if you're unlucky in this 300 base pair region, you have no read. You would estimate that background to be 0, but then from that 1 KB, it happens some other nearby sequence have more reads, then you get a better parameter, then you can use it. And so using this approach, you can see, in fact, most of the regions that eventually will be called a peak, we don't really actually use the genome background. We only use, most of the time, the input control can give us a good idea what's the background should look like. And nowadays with ChIP-seq, when people are sequencing more, even if they don't have the input control, they only do the chip. If we're interested in this region, we just consider this region as the foreground in the chip, and then hollow out the same sequence, and then look at 1 KB, 5 KB, and 10 KB. Just without the center region, we can still estimate a background. And you can also use that to see whether this region is a peak compared to its neighboring region. And so it turns out this approach works even better than negative binomial distribution. So there are algorithms later on that's what developed, which use negative binomial distribution. But because of, so what is a negative binomial distribution? It means it's a Poisson distribution, but in different locations, you use a different lambda parameter. And that's exactly what we are using, right? It's every location we're doing a Poisson distribution, except that this lambda parameter is dynamically estimated based on the local structure. If there are sequencing bias, if there are chromatin bias, it's all factored in. And turns out this captures the local background much, much better. And we use this conservative approach, not just looking at 300 base pair, but 1 KB, 5 KB, and 10 KB, then take the maximum to get a conservative p-call. And that actually works very well, and people are still using it nowadays. And so with ChIP-seq, there are a number of important quality controls. By the way, even if your ChIP-seq is not working at all, you will see peaks for sure. And there are always some peaks. You can imagine some random garbage got stuck on the tube, and then in PCR they get amplified a lot. You will give you a peak, and you always see that in genome. And so how do we know whether the ChIP-seq is really good? Yeah, so the very first QC is without the ChIP-seq. You just, any read you can do fast QC. When you you get the read back to the genome, you do look at the percentage of reads that are mapped to the genome. So the higher the better. We would usually expect at least 50% of the reads mapping to the genome. This is just BWA. And before we even do peak calling, there is one important factor that is useful. This is used by big consortiums called, I forgot, bottleneck something. You can look at this. This is, if you look at, so if we, in ChIP-seq, usually most people care about read that can only find one location in the genome. If this read happens to be in a repeat sequence that can map to 100 locations in the genome, it usually gets thrown away. Right? So you just look at things that can be uniquely mapped to the genome. And at the end, you ask, how many total of those locations do all of the reads land in? So supposedly you have 20 million reads. After each you do the mapping, some reads that are mapped to many locations, they are thrown out. The reads that can be uniquely mapped to one location, let's just say 15 million. So okay, then you look at those reads, you ask how many locations do they map to? You might notice that this read is only finding one location, but there are multiple of the exact identical reads being sequenced over and over again. That's actually not a good situation. And so we look at how many of those locations have only a unique read, only one read that lands there, divided by the total number of locations where a read is present. So basically, in the early days, when people were trying to do ChIP-seq, say in a small tissue, they don't have enough cells in development, in some organ tissues, biopsies. They don't have enough of the DNA. They have to PCR amplify a lot. And in that process, during that process, you will be amplifying the identical sequence over and over again. And so basically, when you have the redundant read, that's not a very good thing. It indicates that you just didn't have enough starting material. Instead, what is good is within this region, you have many, many reads, but they all start at different location, right? So some, basically, this is good. You can see they do form a peak. They all map to around this region, but if you look at where the actual alignment happens, they're all in different locations. That is a real binding. But if everybody is aligned to the exact same place, that means it's a PCR amplification. And so that's an important QC measure. And in fact, when you run MAX for peak calling, MAX will eliminate all of those redundant reads. The same sequence you read over and over and over again, and you map over and over and over again. That's not good. So we eliminate those first before we call the peaks. And then each peak we do a Poisson distribution, the Poisson compared to the local lambda estimation. Do we need to do FDR adjustment? Think about RNA-seq. We are trying to compare condition 1 with condition 2. We ask, is this a differentially expressed genes, right? You have to test every gene. But look at the human genome. You have to test every window to see, is this region higher in the chip compared to the control? That's a lot of multiple hypothesis testing, right? Absolutely, you need to do FDR. That's why you always have peaks, but those could be, you will know with FDR whether those are real or not. We do use Benjamini-Hochberg. in order to reduce the total number of tests, we really do not want to test every possible 300 base pair window in the genome. And so instead, we look across the genome. If this region have too few reads, it's hopeless, right? The RNA-seq does the same thing. If the gene is not expressed in many samples, you just can't use that. If you can remove enough of those hopeless windows, then your FDR, you can actually call more correct signals in there. And so we say, okay, within 300 base pair window, you need to have a minimum number of reads before you even think about comparing with the control. And then you still test the ones that are promising, and then based on the distribution of those p-values, you can do Benjamini-Hochberg to see the correct number of binding sites. Okay, so that's a QC. There are additional QCs. By the way, ChIP-seq is much harder to do than RNA-seq. I remember in the early days when we were doing ChIP, it usually takes a postdoc 3 to 6 months to do, and only a third of the labs that collaborate with us can even get this experiment working. Nowadays, with more and better antibodies validated, better standard protocols, and also sequencing have better signal to noise, I would say maybe majority of the ChIP-seq is okay, but there are plenty of published high-profile papers with garbage data. It's just a fact. So other useful QC is the number of peaks with good FDR and the fold change. We usually cut off by fold change of, you can do either 2 or 5. 5 you will be pretty confident. 2, I think in the early days you can do 2. But nowadays, when people are sequencing a lot, you can use 5-fold. By the way, the total number of peaks you detect they increase with sequencing depth. This is similar to RNA-seq. You can imagine if you sequence super, super deep, even if the gene expression is slightly different, you might be able to detect it. But if you sequence shallow, or if this gene is short, then your power is limited. And so supposedly within this region, the control is 10 reads. The treatment is, the ChIP is 12 reads. 12 over 10, you can't really call it. But 120 over 100, you are a little bit more confident. And then 1200 over 1000, you are much more confident. There is some enrichment in there, right? So the more you read, the more peaks you get. Therefore, in addition to p-value or FDR, you do want to use a fold change to cut out. It's not uncommon for ChIP-seq to have thousands of peaks or tens of thousands of peaks. And so it's actually a good indication. I would say if you do a ChIP-seq and if you cut off by 2-fold and with FDR5 and you only have 30 peaks, that experiment is probably not working. It's very rare for a transcription factor in the human genome to only bind to 30 locations in the genome. They are naturally binding to thousands of locations in the genome. Another useful indicator is called the Fripp score. This is the fraction of reads in the peaks. You remember we say when you do the ChIP-seq experiment, there are some random things that is just non-specific binding to the antibody, or they are stuck on the bead or the tube. It's fairly often. And so at the end, after you call the peaks, you say, okay, I do 2-fold change FDR, I call 3000 peaks. You ask how many reads really land in those peak locations divided by your whole genome, all the sequences that you have done. Depending on the factor, for transcription factor binding, the Fripp is usually 2% or 3%, meaning that most of the reads are just random noise in the genome. Occasionally you see enrichment, right? 2%, 3% of the reads that really land in the genome. For later on, we'll talk about histone modification. You might get as high as 10 to 15%, but you can see with ChIP-seq, you'll be reading a lot of junk before you actually read the sequences on the peaks. However, those nearby random sequences can help you estimate how much the local biases are. So they are still definitely useful. So don't worry about those remaining 98%. They don't land in peak, but they can help you estimate your lambda parameter. So it's still useful. Okay.
Does that
Yeah,
mean
Kevin.
90 does that mean
Sorry,
98%,
Patrick. Huh?
Does that mean 98% background?
Very often ChIP-seq, 90 to 95%, yeah, as high as 98 to 99% of your reads are not on peaks. Yes, Kevin.
So just to elaborate slightly on that, because they don't, because they're random and they're not overlapping, enriching in any sort of fashion, that's what you can
Yes, they are not. Yeah, in the other locations, I mean, 2 to 3%. You're talking about, say, in a 1 KB region, there are still 3 reads or 5 reads in there. But because the human, I mean, think about it. For in the cell, there are how many each cell? How many copies of that transcription factor do you have? Not too many copies of that protein, right? And they have to find a few locations to bind in the genome, whereas the genome is 3 billion base pair. And even if the antibody was to amplify your signals 1,000-fold, that's still 1 in 3 million, right? So signal is much fewer than the noise. The genome size is your noise. Yeah, so that's why ChIP can kind of enrich enough of the signal so that in this case, your signal to noise is about 2%. Yes, because if without the antibody pull down, your transcription factor binding is this 12 base pair sequence out of 3 billion. It's huge. I mean, the signal is tiny. It's a needle in the haystack, right? And then we do also look at the evolutionary conservation. We say, even though if you do a ChIP-seq experiment, you look at your few thousand peaks, and you look at the phase conservation score, in fact, the majority of the binding sites are not conserved. And yet, if you collapse all of those, you align all of the peaks by their peak center and calculate, like 3,000 peaks. Each peak has, by the way, phase conservation score is you have a number at every nucleotide in the human genome. And you say, okay, well, this peak there's one peak, you look at underneath the phase conservation score. The next peak, you look at the phase conservation score. The third peak, you look at the phase conservation score. And you align all of them together, even though majority of the peaks are not conserved, you average their conservation score, you should see a conservation. This is a real transcription factor that has a function. It will bind to at least some of the sequences that are conserved. So overall, when you average out everything, you should see a peak. If this region, you don't see a peak, or you see some really jagged things, you see fewer peaks, those are all indication this data is not so good. Okay. Then you can also look for motif enrichment. If you're doing a GATA3 ChIP-seq, at the end, after you called all the peaks, do you see a GATA motif enriched in the data? GATA is enriched in GATA. That's why it's called a GATA transcription factor. And so the fact that you find the correct motif is also an important indication that this experiment is working.
Can you expand again what
the conservation plot?
Yeah.
Oh, okay. So remember, some people ask for the clarification on the conservation plot. So remember in the ChIP-seq, we call, say, 3,000 peaks, right? Each peak, we know the beginning, the end, and every base on this, you go to the UC Santa Cruz. Actually, we usually download a local copy. Every single nucleotide in the human genome is assigned a conservation score. And so you calculate, so you look at the peak center, you say, what is the conservation at this base, the next base, the next base, the next base. You can calculate, you can just extract that information out. And the next peak, you also look at the center. You look at the nearby nucleotide to look at their conservation score. And the third, you look at the peak center, you look at the nearby conservation score. And then when you look at all of the peaks together aligned at the center, you can calculate their average conservation. The average conservation score should give you something like this with the center of this peak, because that's where the transcription factor really binds, right? Overall, it should bind to more evolutionarily conserved regions. Even though if you look at your 3,000 peaks, you ask, you look at those scores, what are the scores? There will be a lot of regions with pretty low scores. Majority of those peaks are not in very high score regions. But the average, when you align all of the peaks together, the center should be more conserved than the nearby regions. right? Because that's a functional region compared to some random noise in the background. If you don't see that, then that's not a good ChIP-seq experiment. Okay, and then there are other experiments which give us, so by the way, ChIP-seq, this is one transcription at a time. So if I'm interested in how one particular transcription factor, E2F, P53 is binding to the genome, you use one antibody to get it out. And if you want to do the same cell, another transcription factor, it's a different assay. And later on, we will talk about this in future lectures about the DNAs. This is an experiment which can give you almost all the transcription factor binding sites in the genome. They just know something is binding here, but they don't tell you what factor is binding there. But there are assays like that. So if your ChIP-seq data has good overlap with all the DNA peaks, then that's a good indication it's a good quality experiment. Okay, and so in order to streamline this analysis within our lab, actually this has been around for many years now. Sorry, we haven't updated this for a few years as well. We have something called the cistrome.org. Cistrome stands for all the cis element bound by a trans factor at genome scale. Basically, all the DNA bound by the transcription factor in a ChIP-seq experiment. And analysis pipeline is like this web server. Everybody can register for a free account. And on the left side, there are a lot of different tools. And you click on a tool, you can upload some files. You just say, I want to upload the file. And all the files you upload, as well as the processed result, are on the right side. And if you click on a tool and you say, okay, I want to, you can specify, I want to use this file and that file and that file and the following parameters to execute, it will calculate directly on the server that our lab has to the public. And then you can go home and sleep. Next morning, you go back, log in again, that result file will be an additional file underneath. And you can use it for some other downstream analysis, which is helpful. I think for this course, you guys should learn how to do this in Python or you run command line. In addition, we have collected, we have the largest collection of published ChIP-seq studies. And they're also a very detailed QC. You can go to that and look at each of this QC measure. If it's green, it's a good quality data. If it's red, it's not so great. You can see here. This is a study that's published in Nature, and they are doing, but this is because it's an unusual mark. If you look at the signal, didn't quite pass QC. So there are definitely high profile papers that has a few bad quality data. And so if you're interested in, I'm just interested in some transcription factors binding in some cells, you can search for that and then look for the high quality data. And you can use that data to help you understand gene regulation. Okay, questions? So ChIP-seq downstream analysis. After, the reason people are doing ChIP-seq is they want to know what is this transcription factor binding to, and what does it do, right? So there are a number of these interesting things you want to do. The first is you want to look for a motif. In fact, if you have ChIP-seq motif analysis, it's much easier. It's also a very important QC. You can run MEME, you can run other method. Yeah, so we have our own motif finding algorithm, which is used to find motif in all the public ChIP-seq data that we collected. In addition to know that the motif should be enriched in your data compared to the genome background, what we also know is that this motif, if you look at your peak, fold change in terms of enrichment, this motif should appear more often in the peaks that have higher fold change than lower fold change. It kind of makes sense. If that region has 10 copies of the motif, you do ChIP-seq, you'll have multiple transcription factor binding in that location, then you have a higher fold change. In addition, if your peak, you remember your peak is like this, very often the motif should appear more likely to be in the middle compared to the side. because the site is just some random garbage, right? The transcription factor is usually binding right at the peak. And so we do use these two considerations to help us decide which motif is really good. And that tool is already in Systrom. And one thing we notice that the transcription factor in the same family often have similar binding motifs. And but you can also use the ChIP-seq motif to find transcription factor interactions. For example, this is the example for the same TF in the same family have the similar motif. You can see here, this is the transcription factor name. And this is the, if you click on each of them, they will show you the matrix, the sequence logo. And you can see all of them, the TF is different. JunB, Jun, MAF, TBX, Bach1, and FE2. And they are, this one is a little strange, but all the rest are in the same leucine zipper family of transcription factor. And if you click on each of them, their motif look quite similar. But you can use ChIP-seq to look at transcription factor interactions. For example, supposedly this transcription factor 1 is interacting with another transcription factor 2. And this interaction can happen, the 1 and 2 are binding right next to each other within the same module. Remember, cluster of binding that can happen within the same 150 to 200 base pair region, or it could be a big loop, like it binds to 1, and then there's like some other loop on the DNA, and then the transcription factor 2 is binding far away, but because they interact, in the cross-linking step, you will cross-link the TF1 to its DNA, TF2 to its DNA, and also you will cross-link the TF1 and TF2 together. So it will form a chunk together. And then when you sonicate, this will be sonicated into shorter pieces. And so when you sequence the data out, you might notice, for example, in this case, within this peak, it has the motif of TF1. It also has the motif for TF2. But in this case, one peak only has, because you use this antibody to drag down, this piece of DNA will be enriched, but this piece of DNA will also be enriched because they're all together. And so you might see that if you do ChIP-seq of TF1, there are some regions that only have TF1 motif, but there are also regions that only have TF2 motif. And that is an indication that these two transcription factors work together. Okay, so you can look at this from two ways. The one is on the system, there is something called toolkit. If you just enter the ChIP-seq data of one transcription factor, it will automatically tell you what other transcription factor ChIP-seq has a peak that overlaps. So you can imagine if TF1 and TF2 collaborate or they interact a lot, whether you do TF1 ChIP-seq or whether you do TF2 ChIP-seq, their peaks will have a lot of overlaps, right? So, so the system database have the system toolkit. You enter some ChIP-seq data, it will pull out all the other ChIP-seq data that have significant overlap with your data of interest. And that can help you find out whether something overlap. For example, if we do NANOG ChIP-seq, at first we'll say, there are other NANOG ChIP-seq that look really like this. But also you will say, SOX2 also has significant overlap with your factor because SOX2 and NANOG, they interact a lot as transcription factors. Okay, so you can look at these type of interactions based on the peak overlap. You can also look at this directly based on the motif enrichment. So for example, if we were to do this is actually a NANOG ChIP-seq, you will see that the NANOG motif is here, but you also see the SOX motif in here that are enriched, right? So you can see it's a SOX1, sorry, SOX4, SOX11, and SOX10. They are all actually SOX motifs because the same family member, right? So you know that the NANOG family of transcription factors and the SOX transcription factor, they interact with each other. Okay, yeah, yes, Richard.
Sorry, I think I missed the first case. How do you see an overlap? they're distant on?
Oh, those two sequences can be far apart, as far as a few kb or 10 kb or 100 kb probably even. It does happen. Yeah, so sometimes when you do a ChIP-seq, you will see that there is a motif. Your TF's own motif does not appear in some sequence. Instead, another transcription factor motif is significantly enriched in your data. That's an indication. Well, there's also a possibility that you use the wrong antibody. Sample swap, right? But hopefully you find, yes, my factor motif is enriched, but then there is another motif that is also enriched, usually a secondary motif. And that's an indication that these two factors interact with each other. But remember, so here you can see if we do the NANOG ChIP-seq, yes, on the very top we see the NANOG motif, but then you see the SOX motif. You see SOX4, SOX11, SOX10, but there's a SOX2, SOX3, right? Which one is the one? Very often it's the one, is the SOX that's the highest expressed. That's interact in this cell type, that's interacting with your NANOG of interest. Okay. Yeah, so either by a peak overlap or motif enrichment, you can figure out whether some family of transcription factor can interact with each other. Yeah, and then to really figure out which member is the specific member in the family is interacting, usually you look at expression. Yeah, so first of all, this transcription factor needs to be expressed fairly highly in the cell you are interested in. And so do I have an example? Oh, we do have an example. We do have some time. So in the very early days, this is a study that we conducted with our closest collaborator, Miles Brown's lab, where they are interested in estrogen receptor binding in breast cancer. So majority of female with breast cancer have an overactivated transcription factor, estrogen receptor. And you want to ask, where does it bind in the genome? What do they do? And so this is actually in early ChIP days, we got 3,000 peaks in the genome, and reviewers were like, gosh, 3,000, you know, now it's like easily 10,000. At that time, we were able to validate, yes, those binding sites, you can experimentally validate there's binding enrichment. And then we submit that paper initially to Nature. And then Nature just said, oh, you just show that the transcription factor binds to the genome in those locations. There's no insight, right? Is there anything interesting that you can find? So our collaborator came to us to say, what else can you find? So we started looking for motifs, and we said, you know, in addition to estrogen receptor motif, there's another factor, which is in a FORCAD, is a family of transcription factors with a different motif. And so, but then which FORCAD is the relevant one? So we look at the many breast cancer data together to ask, out of all the FORCAD family members, which one is highly expressed, and also the expression is correlated with estrogen receptor? Because it's like these friends have to hang out together to do something together, so that their expression are usually correlated. And once we do that, FOXA1 came on top. And then our collaborator later on was able to show that when you knock out FOXA1, estrogen receptor can no longer bind to their location anymore. And so the FOXA1 not only is an important factor, it's kind of an essential factor for estrogen receptor to bind. And then we were able to get it published in Cell. So you can see that's how informatics really helped the experimental work to generate a useful hypothesis. Okay, and so that's how you use it to find important interaction partners. So ChIP and ChIP-seq was very early days performed on yeast. At that time, you probably remember yeast is about 50% coding. The gene is like 1 to 2 kb long, and then the sequence in between the genes are usually also about 1 to 2 kb long. And so very often, the sequence right in front of the gene regulates this gene. And so you do ChIP-seq, you get a target. It's very easy, right? If it binds in front of the gene, yes, it's going to regulate that gene. But then once you look at the transcription factor binding in human or mouse, this is what you normally see. So a core promoter is only 1%. This is like 1 kb from the transcription start site. In yeast, we only look at 1 kb. In human, if you look at 1 kb out of all those binding sites, that's only 1%. If you look at maybe 3 kb, you will probably get another 3% binding site. And there are some, so 48 is in introns in the gene. And also, if you look a little further in front of the gene, intergenic is just between genes. There are big gaps between gene sequences, right? If you look at things that are less than 25 kb away, that's another 8%, but there are also a lot of intergenic sequences that are over 25 kb away from any nearby genes. And so there are a lot of these in the middle of nowhere introns or intergenic sequences that where most of the transcription factor binds. So right now, it's still really an open question. How do you really assign the target of a gene given that this transcription factor binds to 10,000 places in the genome, right? So if you say, okay, I would say in the early days when people just did the ChIP or ChIP-seq, they say, okay, anything that has a binding in the promoter, you'll get some result. You get, a few hundred genes that has a binding right at the promoter. You also have to ask yourself, well, for promoter, do you mean 1 kb, 2 kb, 3 kb in front of the gene or a little bit inside the gene? Because it's from transcription start site, it's before and after, right? A little bit. How do you define this? It's kind of an open question. Other people also try to assign a peak to the nearest gene. So for example, if I have a peak and that gene is 10 kb and this gene is 5 kb, it's going to be assigned to the 5 kb and not the 10 kb, right? So people also do that. The distance to the nearest gene transcription start site. Later on, people also notice that the number of binding sites near the gene for a transcription factor is also important. You can imagine if you have one gene and nearby you have just one, say, estrogen receptor binding site. Another gene you look at nearby, there are like 10 binding sites. Usually we would believe that the one with 10 binding sites are more tightly regulated by the TF of interest. Whereas if just with one, we're not even sure whether it's working or not. And if you want to use other knowledge, you can still do that. This is still an open question. And I can tell you some general approach that my lab use, and I think it would work most of the time. I mean, it's, I think, as good as any other method out there. So binding, it will tell you which transcription factor is likely, which genes are near this transcription factor binding. Let's see. So if you have a binding site, whether it's right next to the gene versus really far away, there's a difference. If it binds right close to the gene, it's more likely to have an effect. When it binds so farther away, it's not as important. So far, what we have found that is for most of the transcription factors, their binding usually is the distance from transcription start site. Whether it's upstream or downstream is mostly symmetric, the effect. But then, whether or not that binding will really have an effect depends on whether its collaborators are available. So supposedly this guy likes to play poker, but he's only by himself without the other 3 friends, well, then it's harder to play, right? So the collaborating transcription factors are important. And we also need to look at the gene expression. If you compare the situation when the TF is active versus inactive, and you look at the genes that are differentially expressed, that also give you an idea that this TF is important. And so in terms of modeling the binding effect, we will have a later lecture directly on chromatin interaction looping. But just to give you a general idea, people notice this potential exponential decay, which means that if the binding is very close to the gene, any gene, then that gene is more likely to be regulated by the transcription factor. But if this is for interactions, but if something is really far away, they are less likely to even loop into the promoter or the transcription starter of the gene, then they are less likely to regulate. And so this type of chromatin looping is important And the looping generally have this exponential decay characteristic. And with that, based on the binding, we try to assign basically what we call a regulatory potential, which is the sum of binding sites weighted by the distance from transcription start site. So for example, if I want to know what is the regulatory potential of this transcription factor, if I know it's ChIP-seq in terms of regulating this one gene that I'm interested in, I can look at this, so we can have this exponential decay idea. So here, we look at the transcription start site of this gene, and then every time we see a binding site, we ask, how far is that binding site from the transcription start site? And this is weighted by this exponential decay, which means, for example, if the decay distance is 10, actually for a lot of transcription factors, we use a decay distance of 10, which means that if a transcription factor binds right on the transcription start site, that counts as one binding site. But if it binds 10 kb away, it will only be half. And then if for every distance, it decays even more, it's exponential. And so you can sum all the binding sites weighted by their distance with this exponential decay to say, based on the binding, how likely is this gene a target of the transcription factor you are interested in? Okay, so that's just on binding. And that will give you a rank ordered list. This list you can directly download from Systrom. Based on just binding, we will tell you, what are the likely targets of this gene from the top to the bottom. And this is usually, if you see a 2.1, 1.4, 1.4 would probably mean one binding near transcription start site, another one maybe a 10 kb away, something like that, right? And the second one is to look at the gene expression. The gene expression can be based on two different things. Supposedly, so we developed this algorithm a long time ago. If you have a lot of people come to our group to say, oh, I have two cell conditions, one with the transcription factor active and one with the transcription factor inactive. And then we also did a ChIP-seq when the transcription factor is active. We know where it binds. Can you tell me what it does? And so based on the binding, we can calculate what are the putative target, but also we need to look at the expression. We say, okay, when the TF is active or inactive, what are the differential genes, right? So you know which genes are upregulated, which genes are downregulated. You can also look at the gene expression correlation in the tissues or tumors if you don't have differential expression. You just ask what other genes are correlated with your transcription factor of interest. And that will also give you a rank ordered list. And based on those, at the end, we say the rank 1 times rank 2 will give you the final rank ordered list of the direct targets of your interest. And by the way, in terms of transcription factor regulation of gene, it's not black and white. Every transcription factor has an effect on every other gene. It's just more effect or less effect. It's a continuous value. And so at the end, this rank order is also continuous. You say the top ones are the potential direct target. The remaining ones are weaker and weaker and weaker. And you can take the top ones. So in fact, most of the, I would say, minority of the binding sites is functional. You might see 10,000 binding sites, but it only regulates probably a few hundred genes. And at least the strong ones, the important binding sites are probably in the order of a few hundred to a few thousand, a couple thousand. The remaining ones are just kind of weak. And once you take the top target, do the rank product of the two, one from the binding, the other from the gene expression change, you get the top ranking genes. You can do gene set enrichment or gene ontology. You ask, okay, what is this transcription factor's direct targets do in that cell? And then, by the way, this is now all done with one web server you can use, which will be in homework 4. Okay, so that will give you the target. So yeah, in summary, basically ChIP-seq, you can use it to identify genome-wide all the in vivo in the cell, what the transcription factor is doing in terms of binding in the genome. You can do use MAX for ChIP-seq calling, And there are many, many different QC parameters. There are conservation, FRIP score, FDR, fold change, to help you figure out and also look for motifs. But then once you have the ChIP-seq data, you want to look at transcription factor motifs. And from there, you might be able to find that it's collaborating with something. And also, you want to look at its target. By the way, sometimes a transcription factor can have effect both upregulating gene and downregulating other genes. And that is because they are hanging out with different friends. You know, with this group of friends, they will do good things. With this group of friends, they'll do bad things, or they'll have different functions. And so that can happen as well. But once you figure out the target genes and the collaborating factors, you can also look at the gene ontology of the target genes and then do pathway analysis. Okay? Yes?
Here's again, how can we rank genes based on binding?
So let's look at this situation. We look at every gene and look at all the binding sites within 100 kb from this gene, right? And if a binding site is right as transcription start site is weighted 1, it's binding further away, then it's weighted less and less and less. And at the end, we add up everything weighted by the distance to get a score. And so what kind of gene will get a high score? They have more binding sites, and those binding sites are close to the transcription start site of that gene. They will get a good score. They will be the higher ranked target. Whereas the gene has either no binding sites or there's some weak binding sites that are really far away, then they will have a very low score. Okay. One thing I forgot to mention that the decay rate, you know, what is the decay? What we found is that there are general 2 types of transcription factors. One type only have very short range effect. They have effect usually within only 1 to 2 kb away from the binding site. And you can see that as well. Their binding have probably 20 to 30% really bind at the promoter. It's still a minority, but it's like 20 to 30. Or even 10% is kind of rare. Whereas some factors have really wide range remote control type of transcription factor that have 10 kb or sometimes even longer. Their binding site, you look at genome-wide, there would be like 2% binding to the promoters. So just looking at the binding distribution, at least our group can give you a rough estimate of what that decay distance should look like. Okay, but these tools are all available now. You can use. Okay, great. That's good for today.